Okay, thank you so much for the introduction. And the title's already been read, so I won't go through that. The shortened title is Getting the Brain to Take Its Medicine. And just a few disclosures to cover. So this article was published in April last year, and it's suggesting that rather than everybody focusing in on immunotherapy, which we all know has got many, many problems, the next thing to now focus on is drug delivery into cancer cells, which all of those working with aptamers that have been doing this for 10, 15 years all cheered when we saw this article. So we know that doxorubicin, a classical chemotherapeutic drug that's been around for 60 years, does actually kill cancers. The issue is it doesn't kill all of the cancer cells, and that's because the drug efflux pumps on the cell membrane. But if we can find a way to get around that, then we can give this a much more therapeutic treatment option to patients. And that's where the aptamers come in. If we can get the drugs far enough into the cell, away from the drug efflux pumps, then they cannot pump the drug out, and the drug will then go to the nucleus and kill the cell. And typically that's through receptor-mediated endocytosis, where you bind to a receptor on the cell membrane, and that's then internalized through endosomes and lysosomes, and then the drug is released and it goes into the nucleus. So the target that we first looked at was EpCAM. It's the very first cell surface marker that was recognized on cancer cells. It was identified in the late 1970s. We know the antibody therapeutics have shown some efficacy, but there's a number of caveats as to why monoclonal antibodies are not really effective. And it's only really effective if you've got extracranial disease. And so I've tried to highlight some of the students that have been responsible for doing this work. This is Dongxi Zhang, who during his PhD demonstrated that an RNA aptamer targeting EpCAM was effective. We showed it was really stable at various different pHs until you get to the pH of a lysosome, where the DOX is protonated. It's then released from the aptamer structure and can then carry on moving into the nucleus. And we did some biodistribution, and that was looking at the biodistribution 24 hours after a tail vein or IV injection. And we also showed that we're able to treat the cancer cells ex vivo, put them into the animal, and this is panel A. The tumor cells that were treated with the aptamer dox prior to implantation didn't really take off and grow a huge amount compared to what we'd seen with all the various different controls. And then in B, we showed that the aptamer dox significantly extended the lifespan in a xenograft model of colon cancer. And that blue line then extended way further out until we had to cull the animals for cost reasons. In the bottom left panel, we showed that the aptamer was able to penetrate much further away from the blood vessel into the tumor as compared to a monoclonal antibody. And again, the biodistribution. But the one thing that we could not do was get into the brain. And we know that this is an important factor because a number of patients that are presenting with brain mets or with brain cancers are increasing, even 5, 10, 15, 20 years after the initial treatment, which did show success because we're not killing all the cancer cells. And so we can see here some of the plots of showing the increase of metastases spreading 5, 10, 15, 20 years after initial diagnosis. And we know that getting into the brain is a bit of an issue. 98% of all drugs do not cross the blood-brain barrier, and we need to have a much better way of getting in that doesn't involve completely disrupting the blood-brain barrier to get drugs in there, because you'll then get all the toxic substances that are floating around in our bodies also getting in there and causing neurological conditions. And so what we did was we used the same method that the drug uses to get the essential drugs, the essential proteins and things into the brain, the transcytosis. But we also knew that getting into the brain was not going to be the only thing we needed to do, because if you just get drugs into the brain, they're going to go wherever they want and are then going to cause neurotoxicity. So we developed a bifunctional aptamer, one that binds to the blood-brain barrier, it binds to the transferrin receptor, and then we added a DNA aptamer that targets EpCAM. So we've got the two arms, one that crosses the blood-brain barrier, the other that binds to the cancer cells, and then we can integrate doxorubicin using its mechanism of action of binding into GC base pairs. And so this work was done by my PhD student, Joe McDonald. We tested in a healthy animal model first, because we know the blood-brain barrier is completely intact, and we demonstrated that we could get into the brain. And we also tested this in a triple negative breast cancer brain metastases model, and demonstrated again we were getting into the brain and we were getting into the cancer cells. And then we saw that by pre-blocking the EpCAM on the systemic tumors, those below the neck, we were able to get a tiny little bit more into the brain to target the cancer cells. I say a tiny little bit more. There's a lot of work here that was truncated because of the pandemic, and we're just slowly picking back up again now. I'm obviously from Melbourne, and we were significantly hit by that. But when we looked at the attachment of the drug into the aptamer stem, we saw we were getting just that little bit more drug getting into the brain. We saw that the binding affinities were not greatly changed, a huge amount for some of the aptamers. I will point out that the binding affinity to the transferrin receptor on the blood-brain barrier was unchanged in what I call our positive control. In our negative control, the TENN, the docs actually stabilized the structure to the extent where we actually had a binding affinity to the transferrin receptor. And I'll come back to that one shortly. The binding affinity to EpCAM with the docs intercalation in the pos control did actually decrease to about 500 nanomolar, but we think that that's actually been beneficial when we're developing the therapeutics. And you can see from the images on the right, we've got the GFP labeled tumor cells in the brain, and we did see some localization, which is much better on the next slide. But with regard to the half-life, the docs also stabilized the structure. We went from a half-life of the bifunctional aptamer with no drug to be about 10 hours, to then double that half-life to about 22 hours once the docs was intercalated into the stem. This shows the in situ slicing of the brain tissue with the tumor GFP labeled. All of the black around that is the healthy brain tissue. We can see that we've got aptamer in the GFP labeled tumor. We can see that the docs is in the GFP labeled tumor. And then when we zoom in even more, we can see that we've got drug inside the cancer cells within that tumor that has spread to the brain. And this is a intracardiac injection of the cancer cells, so they naturally invade into the brain. We're not disrupting the blood-brain barrier while we're introducing the cancer cells, or not to the extent, or not more than the extent to what it would normally. Now, we've still got a fair bit of optimization to do on this. We saw that the aptamer docs did decrease some of the tumor burden in the brain. What was much more significant and surprising was that we significantly decreased the tumor burden in the bones and the ovaries. So going back to the work we did with the pre-targeting of just a single EpCAM aptamer, if we block some of those systemic sites around the body, then we should be able to get much more into the brain. And that is work that we're currently looking at completing, and we'll hopefully have much more results by next year. But even reducing some of that burden in the brain, when we know that there's a number of different therapeutics that have been heralded as being good and then don't work when they go into phase 2 or phase 3 trials, means that we could have something really good here that we just need to optimize a little bit more before going into clinical trials. So part of the title for this was developing a platform technology. So we've now seen 2 papers independently validate the transferrin receptor aptamer at delivering cargo into the brain and increasing the amount in there. The one on the left is a tau aptamer that's being connected to the transferrin receptor aptamer. And then we've also got the transferrin receptor aptamer delivering a protein payload into the brain. So we've got two different things that we're looking at now, the platform technology, as well as the therapeutic that can cross into the brain. So then going back to what we called the negative aptamer, that had the exact same structure as our positive aptamer, but just the base pairs within the binding loops changed. This is work that Justin Henry did during his PhD. And we saw that even though the TENN aptamer had a binding affinity of about 850 nanomole to the transferrin receptor, we used this as a kind of control for the experiments, and surprisingly found that this, what we call still a negative aptamer, was able to reduce the number of cancer stem cells when we were doing an extreme limited dilution assay. And so we now are looking at the use of both aptamers as therapeutic, because the one takes out the cancer cells that are there, the other takes out the cancer cells that could proliferate and continue to cause metastases in the patients that could occur years later. And then looking again at that transferrin receptor aptamer, we know the transferrin receptor is overexpressed on glioblastoma. So we're looking at using the same aptamer to cross the blood-brain barrier and then target glioblastoma cells. And we've got some preliminary work done by Brianna Giles, who's just started as a PhD student in my lab. We've shown that the efficacy of DOX and also the aptamer DOX is the same. So by putting the DOX into the aptamer stem, we would reduce the neurological side effects that we see with a number of treatments. And we can see that the aptamer DOX penetrates the glioblastoma tumor sphere much, much better than it does with an antibody, which is work that we did about 10 years ago. So just see what the time is. So quickly, what we've been doing during the pandemic is looking at doing selects against other targets. Don't ask me why we chose to do that during the pandemic. But work that Sahara Attenborough has been continuing on from other honour students is looking at developing aptamers to CSPG4, work that we started when we were looking at targeting malignant melanoma, which is a major problem we've got in Australia. But we've then done a lot of other work with CSPG4, and it's overexpressed in quite a number of other solid tumours as well. And so we did the initial fluorescent binding showing that the highest fold change was in round 7. We did the selects. Sorry, I can't show you any of the sequences, but we're working on patenting this at the moment. And we saw that in medulloblastoma, which is a pediatric tumor in the brain, that we had a reasonable binding affinity. We also saw that the aptamer was able to internalize into the medulloblastoma cells, and then work done by colleagues up in New South Wales also looked at the ability of this aptamer to attach to fixed cancer cells using a melanoma cell line, and showed really good efficacy. Of note, there are people that usually work with antibodies and were completely blown away by the signal expression they were seeing with this, even when they diluted it down to a 1 in 400 dilution. So I think I've managed to change somebody's opinion with aptamers. And then very, very prelim work that we've done with another target, CD98. We know that's overexpressed on the blood-brain barrier. There was a study that showed that targeting CD98 gets more cargo into the brain than the transferrin receptor. And again, even though we chose that purely for its transcytosis, it's also overexpressed on a number of solid tumors. So what we're doing is developing that platform technology. We've got the CSPG4, CD98, transferrin receptor, and EpCAM aptamers. All of them can be put into different packages and platforms so that we can then get into the brain or target the systemic mets. And so just quickly summarizing we've got functional delivery agents. Targeted delivery, we all know, improves survival, and we're getting across the blood-brain barrier into the brain and looking at targeting other populations. And so all the people that are bolded, people that have been involved in this project, but I just want to entice all of you to consider traveling to Melbourne and Perth in November, as we are holding our first APAC Optimus conference, and that's on the Monday and possibly Tuesday of the week, followed by the Functional Nucleic Acids Conference on the Thursday and Friday in Perth. So you get to see two beautiful Australian cities for the price of one, and in only 1 week.
Thank you very much.